Okay, so I've been getting a lot of questions about Lab 9 metabolism. I think that you guys are just overthinking it a little bit. I'm going to start with the TSI, the triple sugar iron media. So as you can see on here, you can have a lot of different outcomes. Okay, let me make this a little bit bigger so you guys can see it. But when we inoculate these slants, they're slants with a deep butt, and the purpose for that is that you're supposed to inoculate deep within the butt and then on the surface of the slant, and you're trying to obtain both anaerobic and aerobic outcomes. The anaerobic outcome, of course, inside the butt, and the aerobic one on the surface of the slant. So you can have a lot of different things happening. For example, anaerobically, this middle dark one is an example of an anaerobic event. And what happens is the media has sulfur. And if you have an organism that uses sulfur instead of oxygen as a final electron acceptor, then it reduces sulfur into sulfur H2S. Okay, just the way that oxygen would be reduced in you to produce H2O, water, because oxygen is a final electron acceptor in the electron transport chain. Here, sulfur is the final electron acceptor in the electron transport chain, and it makes hydrogen sulfide gas, H2S, okay? And that H2S then complexes with a heavy organic compound in the media, and it causes it to precipitate and turn black. So that means that this organism right here can reduce sulfur, okay? And that is an intrinsic property of the organism. In other words, every organism functions differently based on their metabolism. If you have a sulfur reducer, that's going to be different than a different organism. So this is sulfur reduction that is seen in anaerobic conditions. If we look over here, we see acidification. So acidification means that the organism has the ability to catabolize the simple sugars in the media. When it does this, it releases protons, which lowers the pH of the media, and it turns the media yellow. Okay. Now, if the organism cannot catabolize the sugar, but can catabolize the protein in the media, then the amino acids in the media are broken down. Ammonia is released. Ammonia is basic in character, and that basic character turns the slant a dark red color. Again, the reason for this is because we have a pH indicator. So another thing that you might see is the ability of an organism to catabolize both sugars and proteins. If that's the case, the entire slant will turn yellow as the sugar is catabolized, and then it'll start turning red. This is called reversion as it finishes catabolizing the sugars and switches over to protein. Um, Again, very characteristic of the organism. So here we're trying to discern the identity of the organism based on its metabolic traits. So I'm gonna pause here for a second, and then I'm gonna let you guys see how this is done practically. Okay. Okay. So this is what the TSI slant looks like. The one that Carolina Biological looks like this. Okay. Triple triple sugar iron auger has a deep butt. I had Chris make extra because I was expecting you guys to have problems with it. So those are the ones that we made here for the lab, same color. Okay. And what do we need? Well, we need the TSI. I think your kit comes with 3 of them because you're going to do three different organisms. So you're going to need your loops, you're going to need your TSI, and you're going to need your 3 cultures. Okay, I'm going to demonstrate using E. coli, a slant. Can you use a broth? Absolutely. Can you use a slant? Yep. Can you get it from a petri dish? Yep. At the end of the day, it really doesn't matter. Now, I'm going to demonstrate with the slant because all the bacteria that I'm handing out tomorrow morning to all of you are going to be in slants. So you might as well get accustomed to this guy. So I'm going to need my little lamp here. Mine's bigger than you guys. That's okay. It's all right. Here's my little lamp, alcohol lamp. And that's because everything that we do here has to be done aseptically. Okay. So what are we going to do? We're going to take out 1 loop from here. We're going to do it aseptically. That means that we're going to push it up individually outside of the package, like so. Okay. Once it's outside, I'm going to grab that loop and pull it out. That leaves everything else nice and clean. Okay. I'm not flaming this. It happens to be in front of my fire, but I'm not flaming it. Okay. Don't do that, or you'll melt it. The reason I have this guy here is because there's my E. coli, and that needs to be flamed. So with the back of my hand, I'm going to grab the cap, just like so. You guys can't see that, can you? How about? There we go. Just like so. I'm going to push my lamp out of the way. Can you guys see that? Okay, the back of my hand, I'm going to unscrew my slant. There it is. And I'm going to pass it through my fire. 1, 2, 3 times. Okay. I'm going to take my loop and I'm going to get an inoculum. I'm not going to break the media. If I break the media, I'm going to transfer agar. That's ridiculous. I don't need agar. I just need the E. coli that's growing on the surface. So I'm going to stick this. I'm going to stick this in here. Hopefully you guys can see that. And go in there. And I'm going to grab an inoculum of E. coli, like so, nice and gentle. Okay. It can barely be seen, but I can see it with the naked eye. It's right there on my nichrome, nichrome. This is not a nichrome loop, on my plastic loop. I'm going to flame my slant. I'm going to recap it. Okay. Now I'm going to grab my TSI. I'm going to use the one that Chris made for me. There it is. With the back of my hand, I'm going to grab the cap. This is very awkward. I don't know if I should do it in the other direction. You guys see that? How I'm grabbing this with the back of my hand? Okay, just like so. And I'm going to unscrew it. I'm going to unscrew it. Take it off. And what I'm going to do is I'm going to stick it deep into the media. We're going to go in here. We're going to go deep into the media. All the way down. I'm not going to twist. I'm not going to do anything because if I twist, then when I pull it out of the media is not going to close behind it, and I'm not going to have an aseptic environment. So I'm going to go straight in, and then I'm going to go straight out. Straight in and straight out. Just like so. I'm going to flame it. 1, 2, 3. And recap it. Okay. Now, That's one. We have to do it again. Okay. So I'm going to grab my E. coli again. There's my E. coli. I'm going to grab it with the back of my hand. I'm going to take the cap off. I'm going to flame it. 1, 2, 3. I'm going to grab another inoculum from it. Barbara's here. She's trying to be quiet, but she's not really quiet. We're going to take a small inoculum out of there. There it is. I'm going to flame my tube. 1, 2, 3. And I'm going to grab the exact same tube a second time. Okay, there's my tube a second time. I'm going to take the cap off. I'm going to flame it. 1, 2, 3. I'm going to go down to the bottom, but I'm not going to pierce the media anymore. I'm going to go just to the bottom, just like that. And then I'm going to fishtail on the surface of the slant, just like that. Back and forth, back and forth. So this is the surface. Okay, just like that. Once I get to the top, at a 30-degree angle, I'm going to flame it. 1, 2, 3. I'm going to recap it, make sure it's labeled with E. coli. And I'm done with that tube, and then I can do the other two. Okay, this goes into 70% isopropyl alcohol in order to disinfect it. And we're going to pause right there for just a moment. Okay, so now we're going to look at activity 2, which is oxidase. Oxidase, it used to be believed that oxidase implied that the organism could reduce oxygen. That is really not the case. What oxidase indicates is that the organism has cytochromes that can reduce compounds in its electron transport chain. So even organisms that are anaerobic and do not grow in the presence of oxygen can come out to be oxidase positive, okay? And conversely, you can have aerobic organisms sometimes come out oxidase negative. And again, this has to do with the presence or absence of specific cytochromes in the electron transport chain. And I'm going to pause here so we can look at the practical aspect of this. Okay, so here we are. We're going to test for oxidase. And for this, we need the oxidase 1 ml, this little guy right here. We need our filter paper. And I'm aware that the author says to cut into 3 parts, but I really don't see why. It's not like we're going to cover the whole thing in bacteria. And we need our 3 cultures. We need E. coli, Micrococcus luteus, and Staphylococcus epidermidis. And one of the questions that I got in a couple of emails is that the instructions say to get these cultures from the TSI slant. Does it have to come to the TSI slant? No, it doesn't. Why does the lab tell you to do that? I don't know. I really don't care anymore. There's a lot of strange things in these lab protocols that I don't understand why they do it. The one thing that we do need to be aware of is that they are relatively fresh cultures of microorganisms. Okay, so I think that the author tells you about 2 weeks. If you're concerned that yours is too old, then just do a new slant or do a new broth. Okay, and once put it somewhere nice and warm. I suggest people to do it on put it in a sandwich bag, a Ziploc that is impermeable, and then put it above their television set, believe it or not. The area above your television set is sometimes it's actually too hot. So you might need to put a thermometer in there and make sure that it doesn't get too hot. But if you put it there, within 24 hours, you'll have a nice turbid broth or a nice, well-defined colony like this one, or not colony, but carpet. I can see it here, so I know you guys should be able to. Look at that. That's beautiful. So anyways, does it need to come from the TSI? No, it does not. Okay. So again, we're going to start with an inoculum. We're going to need these little guys. And we're going to go ahead and take one out. Okay, there it is. And we're going to go to we're going to need to light our little fire here. There it goes. And that's so I can grab my slant. Here's my slant. And just as I was demonstrating before, let me move this out of the way a little bit. With the back of my hand, I'm going to grab that cap, like so. I'm going to twist it. I'm going to flame it. Ba-doop, ba-doop, ba-doop. Then I'm going to put my little loop in there. And you get a heavy inoculum. Okay. That looks really good. And flame it. Recap it. Okay. There's my E. coli. So I'm going to go to the E. coli area. And put it on there. You guys see that? Trying to figure out where the center of my camera is at. Okay, we're going to put it right there. We have to get some more. Okay. That's all right. 1, 2, 3. I can get it closer to where I can see. That's probably more important. Oh yeah, that's nice. And then flame. 1, 2, 3. Cap it. Go to my E. coli. Right about there. Oh yeah, that's nice. Okay, just like so. This is going to go into 7% isopropyl alcohol. And now we're going to take 5 drops of the oxidase test. And I'm going to cheat because I can. So I can use a little pipette dialed at 200 microliters. That's approximately 5 drops. There it is, 200. And grab this guy right here. I'm going to open it. I'm going to take out my 5 drops. Like so. I'm going to put on my E. coli. Just like so. Oops, you guys can't see, can you? Just like so. Okay. You gotta have a timer, and we wait. Okay. Now, what's the positive result for this? Positive result is purple. right? Let me go ahead and share my screen here. Okay. There's oxidase positive, oxidase negative. So if it stays in amber color, that's oxidase negative. If it turns purple, then it's oxidase positive. So we can look at our little guy right here. Okay. What the author did is he tore out the paper and crunched it up so we could see the color better. But I can see mine turning right now. Can you see it, Chris?
Mm-hmm.
What color do you see?
It was yellow from here.
Okay. And that's the gist of this. So that's all there is to this investigation. You do the other one with Micrococcus luteus, and the other one is Staphylococcus epidermidis. I'm going to put my tip in isopropyl alcohol and move on to the next experiment. Okay, the third part is catalase. So again, in the first part of this investigation, we're looking at the way that the organism breaks down sugars and proteins and how its electron transport chain functions. In the second part, in oxidase, we tried to assess the presence or absence of cytochromes and the electron transport chain of the organism. And in the last one, which is catalase, what we're doing is we're trying to assess the presence or absence of an enzyme. Okay. So what is the enzyme? The name of the enzyme is catalase. You and I have this enzyme. How do we know? Well, when we put hydrogen peroxide on a cut, the catalase in our blood breaks that hydrogen peroxide into water and oxygen, because hydrogen peroxide would otherwise be poisonous to us. Does the same thing with bacteria. Some bacteria can defend themselves from hydrogen peroxide by producing catalase, and some bacteria cannot. Okay? And how do we tell the difference? The same way that we tell the difference in us. If hydrogen peroxide is broken into water and oxygen, it's going to bubble. That means that you have the enzyme. If it does not, if it cannot break the hydrogen peroxide into water and oxygen, there will be no bubbling, and therefore you will be catalase negative. That's all there is to it. Okay. So what are we going to do? Here, let me stop sharing for a second so we can look at this. What do we need? We're gonna move this guy away, and we're gonna need the hydrogen peroxide that comes with the kit. We're gonna need the microscope slides that comes with the kit. Okay, and not surprisingly, we're going to need our loops. Okay, so again, we're going to put some organisms on here, and then we're going to add a couple drops of catalase, and if it bubbles, we know that it's catalase positive. So not too bad. So I'm going to turn my fire back on. There it is. And I'm going to aseptically remove one of these loops. There we go. I'm going to take it out. And I'm going to go back to good old E. coli here. Here's my E. coli lamp. The back of my hand. Do you guys see the back of my hand? The back of my hand, I'm going to put it right there. And I'm going to twist it, open it. I'm going to flame it 2 to 3 times. 1, 2, 3. And then I'm going to stick my loop in there. And I'm getting some of the broth at the bottom. That way I know I'm getting some E. coli. It's nice and cloudy. That looks really good. I'm going to flame it. 1, 2, 3. And then I'm going to cap it again. There it is capped. Now I'm going to put it away. And I'm going to put that on my little slide on one side because I only got 2 slides and I've got 3 organisms. So I can't put it in the middle. If I do, I'm going to run out of space to do this. So there we go. A drop just went on there. This is going to go into 70% isopropyl alcohol. And then I'm going to grab my huge bottle of hydrogen peroxide. Now I'm going to turn this guy off. We're going to add a couple drops. Add a couple drops to it. Let it go for a little bit. And it appears that we have a catalase negative organism here. Oops, sorry about that. So no bubbles. The bubbles that come out, then that would be a catalase positive organism. Okay. And this doesn't have to do so much with metabolism. So it's, even if it's an older culture, it'll work because the catalase is an enzyme that's manufactured as a protection by the organism. But no bubbles, so catalase negative for this one. If you don't need this slide, it's going to go into 70% isopropyl alcohol. And that's it. I hope that answers your questions. If not, let me know. And we'll continue. Thank you.